Again, this is just an example. We'll see like how, to what level of detail you need to know this. Probably not much, but yeah. So let's say we have, and so these, you see like these chromosomes are homologous chromosomes. I got one of them from the mother, from the father, one of from mother, from a father. And these are like, you see, like they have the same length and they probably have the
Yeah.
same genes. So let's say there's a
Okay.
gene A and there's a gene B. Let's say gene A here and then gene B here. And let's say again, to make it more diverse, let's say this is big A and this is big B. I'm talking about the gene that is on it.
Okay.
And let's say this is, sorry, small a. And then this big B and then this
Okay.
B. So first of all, we are diploid. So again, these are homologous chromosomes. Again, you're like, how do you know they're homologous chromosomes? These are chromosomes that have the same genes, but they don't have to have the same allele. By the way, they could be totally same allele. Like I could get, for example, from your parents, you might get brown eye color from one parent and brown eye color from another parent. So they both would be, for example, if big B, if the gene B is for eye color, it would be, for example, big B, big B.
Mhm.
They don't have to be different. I'm trying to put them, make them different so you see the difference easier.
Okay.
So, this is so homologous and they're homologous. So, and right now, again, it's diploid because I have two copies of, I have two homologous chromosomes. So it will be
Mhm.
2N, and there's four of them, so four. That's what I
Okay.
got. So before it goes through meiosis, first, all of these chromosomes have to replicate, and you end up creating sister chromatids.
Okay.
So you see, the new one has to have the same allele because I'm making a new chromosome from this one. So it has to be identical to that one. Like you can't have a small a here because where would you get small a from? I'm making this one from this one. So you're
Okay.
replicating, yeah. So it would be small a and then small a and then big B and then big B and then small b and then small b. So again, you're like, okay, so what is the numbers? So you see, the diploid and haploid is based on whether you have homologous chromosomes or not. So by
Mhm.
the way, in terms of terminology, these ones are called sister chromatids. But and again, like you're still considering these as homologous chromosomes.
Mhm. So, okay, so each individual one is sister chromatids, and then, for example,
Each one of
AA
these
and
lines,
small
yeah.
AA is a homologous chromosome.
Yeah, so this is
Yeah.
kind of like how it works is that you'll see I'm gonna write the number here too. As long
Hmm.
as these sister chromatids are together, you count them as one chromosome.
Okay.
So that's one chromosome and that's one chromosome. But when they separate, which will happen later, you're gonna count them separately. But yeah.
Okay.
So you only call things that can come from different parents and have the same genes homologous chromosomes. These are didn't come from different parents for you to call them homologous chromosomes.
Okay.
They're sister chromatids. And
Okay.
these two would be homologous chromosomes. So again, because you still have two homologous chromosomes with each other, you're still 2N, and you're going to consider these chromatids as one chromosome as long as they're connected to each other. So you're still 4.
Okay.
So now we're going to go through different steps. Like, yeah. So again, I'm gonna go over this so for you to have an understanding, because again, they talk about a lot of things, but until you actually see what they're talking about, you don't really understand it. Otherwise, you actually learn to learn about these things, and you have to be able to draw these things like in BIS Toy 2. But I'm saying
Hmm.
this is uh it's better for you to have seen it than like they just talk about it. Because they're going to
Okay.
be tested, but you just don't know what they're talking about. So anyways, so then it's going to go through meiosis 1 and meiosis 2. In meiosis 1, whoa. So this is called metaphase 1. So that's when homologous chromosomes will come and cross each other in the middle. So,
Okay.
so for example, if big A and big A are here, then small a and small a here. So you can't put a B here. The homologous chromosomes are across each other. And these are spindles that kind of moving them around. Now, look, there is something called independent assortment, which we're going to see later.
Hmm.
Independent assortment says that when you're trying to put the next chromosome here, like again, we have two options, right? I can put a big B here or small b here.
Hmm.
There's a 50-50 chance. The fact that there is a 50-50 chance that I can put the big B here or big B here is called independent assortment, because that actually influences which alleles, which genes will go together in the gametes.
Okay.
You will see like later. So let's say we put big B here and small b here. So metaphase M for metal, they're actually aligning the metal.
Okay.
Then in anaphase 1, so A for away, it starts to pull these back.
Move it back.
Yeah,
Okay.
because these are going to split into two cells,
Hmm.
like from the middle.
Okay.
Now, this kind of actually cuts in half. So they turn into two cells.
Okay.
So it will be like this. Yeah.
Hmm.
So now, in terms of number, diploid and haploid, first of all, this is haploid. Why? Because the other homologous chromosome for A is here. So you only have one copy of each homologous chromosome. So it's going to be N, and you consider each one of them as one chromosome. So it would be two.
Okay.
Yeah. So, so then these guys are going to enter meiosis two, each one of them. So
Mm.
again, they're going to have, by the way, this line is kind of just like a show that where the middle is. Doesn't mean anything.
Okay.
So this time the sister chromatids are across each other. So the n is equal to 2 still, n is equal to 2. So, and this is called metaphase 2. Then they're going to separate.
Okay.
Again, A for away. So look, I told you, like we consider these sister chromatids as one chromosome as long as they're together. The moment they
Hmm.
separate, again, and this is, again, they're still part of the same cell. Now there's four of them. But in terms of homologous chromosome, these are not homologous chromosomes. The other homologous chromosome for this chromosome is here. So you're still haploid. But
Still
these guys
haploid.
would be considered as separate cells. So it would be four. And finally, you create the gametes. So these guys turn, so each one of these, they're going to turn into a gamete.
Turns to the can.
See, each gamete is a haploid and have 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 wait have half as many chromosomes as originally you had.
as half as many.
See, you're haploids.